Here we have a DNA sequence of a coding strand. We can take this sequence and create our mRNA sequence. To do this, we simply replace thymine with uracil. Using the genetic code, we can determine what amino acids these codons code for. In this particular strand, we start with methionine, as always, and tryptophan, asparagine, alanine, and cysteine to follow. When we get to UGA, we get a stop codon, indicating the end of translation, so the amino acids proline and glycine are not translated into the protein since they are after the stop codon. Let's look at some mutations. Keep in mind these mutations occur in the DNA, so we will look at how the DNA mutation affects the mRNA and the amino acid sequence. Point mutations substitute one base for another. Two types of point mutations are transitions and transversions. Transitions substitute a purine for a different purine or a pyrimidine for another pyrimidine. A transversion substitutes a purine for a pyrimidine or vice versa. We are now going to look at a missense mutation in our DNA strand. A missense mutation alters DNA, resulting in a codon that codes for a different amino acid. If we look at our DNA strand, we notice there's a transversion from cytosine to guanine on what will be the third codon. When transcription occurs, the resulting codon is AAG, not AAC. AAG codes for lysine opposed to asparagine. A missense mutation can result in a non-functional protein or a protein with a different function than intended. A nonsense mutation alters a codon that codes for an amino acid into a codon that codes for a stop codon, therefore ending translation prematurely. In our DNA, we see a point mutation in what will be the fifth codon. This transversion substitutes cytosine for adenine. Once transcribed, the resulting codon will be UGA, which is a stop codon, and will end translation, making the amino acid sequence short by one amino acid in this case. A silent mutation is a mutation in which a codon is changed to another codon, yet it codes for the same amino acid, so it does not change the amino acid sequence and the same protein will arise. A transition occurred in our DNA from thymine to cytosine, and which will be the fourth codon. The mRNA will now read GCC opposed to GCU. However, GCC and GCU both code for alanine, so there is no change in the protein. A read-through mutation occurs when a stop codon is changed to a codon that results in an amino acid. This leads to a longer protein because translation will no longer be stopped at that specific point. A longer protein can mean a different function. We know TGA in our DNA results in UGA in our mRNA, and UGA is a stop codon. However, a transition occurred substituting adenine for guanine, so our codon now reads UGG and codes for the amino acid tryptophan, so our protein will now include tryptophan as well as proline and glycine.